listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is August 3rd, 2015, and I'm your host, Dr. Jay Portnoy. Our topic today, sublingual immunotherapy. Our presenter is Dr. Linda Cox. She's a former president of the American Academy of Allergy, Asthma, and Immunology and an assistant clinical professor at Nova Southeastern University School of Medicine in Fort Lauderdale, Florida. Our first presenter is Dr. Linda Cox. Dr. Cox is a recent president of the American Academy of Allergy. She's also a recognized expert in the field of immunotherapy and more recently sublingual immunotherapy. I started working with Linda Cox over 10 years ago when we wrote one of the first practice parameters on immunotherapy. That's when I discovered that Dr. Cox is one of those people who just gets things done. And I think that's the secret of your success. When something needs to be done, you agree to do it, and you actually get it done, and it never stops. I don't know how you get all the energy to do it. So we're really grateful to have you with us today. I should also mention Dr. Cox is on the American Board of Allergy and Immunology as well. So without further delay, we'll go ahead and turn the podium over to Dr. Linda Cox. Welcome to Conferences Online Allergy.
Thank you, Jay, for that very nice introduction and for the invitation to speak again to this conference. First of all, can everybody hear me clearly? I know it's a little difficult when we're talking through computers and cell phones, but can you hear me?
Yeah, you're absolutely clear.
Good. Okay, that's my Bose headset. I'm going to be speaking on sublingual immunotherapy. And by the way, the previous slide was my disclosure slide, and really relevant to this discussion. I was on Greer's advisory board, but it's as of November of last year, and Greer has a license to one of the sublingual immunotherapy products that I'll be presenting. But the focus of this particular presentation is to look at the efficacy of sublingual immunotherapy in the context of comparison with standard treatment for allergic rhinitis and asthma, which is pharmacotherapy, with comparison to subcutaneous immunotherapy, and then discuss some of the specific things that I think are important with sublingual immunotherapy that is unique compared to subcutaneous, and that is dosing and dosing regimens. Discuss some of the other kind of unanswered questions, when to initiate treatment based on package insert or based on published literature, and whether it's year-round treatment or what's called discontinuous treatment, or pre-co-seasonal is another term. And then discuss safety, but safety specifically with a focus on the package insert of the U.S. licensed product. I did have to switch my time slot. I was originally scheduled to speak a couple weeks ago, and I changed positions with Hal Nelson, who presented subcutaneous immunotherapy. And I think that that's the right order, because subcutaneous immunotherapy has been in use for well over 100 years, and this is often the standard that sublingual immunotherapy has been held to, as it is a much newer treatment. But it probably is as exhaustively studied as subcutaneous immunotherapy in the shorter period of time that it has been available to Europe and now recently to U.S. As of April of 2015, there have been 84 randomized controlled trials with sublingual immunotherapy. Most of these have been with grass or house dust mites. And most of the recent trials that are in kind of the works now, and you'll be seeing published or in press, are in house dust mite, as there are 2 tablet products that are being hopefully launched in the next year or 2 by 2 large extract manufacturers. And then there's been a smattering of other allergen studies. Paritaria is a weed that's important in southern Europe. And we actually have it here in the U.S. It's called pellitory, but as an extract it's not available. and then ragweed cat, a few alternaria studies, only one of which I think is randomized controlled trees. And then there are many allergens that have never been studied in terms of dose response, such as oak tree or dog. This slide gives you sort of a schema of the history of SLIT, starting in 1986, which was the first double-blind placebo-controlled trial. Very, very short trial, low-dose dust mite with a nasal challenge as the primary outcome. And then over the years, more studies have taken place, and there have been systematic reviews and meta-analysis that have consistently shown that sublingual immunotherapy is at least as effective as its comparator pharmacotherapy, or otherwise known as medications. And then in 2014, in the spring of last year, the FDA approved 3 new sublingual immunotherapy products, two grass products, and a ragweed tablet product. So I'm going to start with the comparison by looking at sublingual immunotherapy efficacy in comparison to standard drug treatment. And what you should be aware of with looking at immunotherapy in general, subcutaneous and sublingual, and I think this is important, is you're looking at trials that generally require a natural exposure. If it's a seasonal allergen, it would be the entire season. If it's perennial, it would be some chunk of time during a year-round study, versus a pharmacotherapy trial that is generally a short-term trial, 2 weeks, maybe in the peak of pollen season, of which the treatment group is compared against a true placebo. So the placebo group literally is not taking any medications for their allergy symptoms. Whereas immunotherapy trials, both SLIT and SKIT, are almost always compared against standard drug treatment. Patients are allowed to take medications, rescue medications, of which these medications are scored, and it's part of the outcomes that are assessed. But this does bring down the symptomatology of the patients in the study, because they are allowed to treat their symptoms with medication. And the other thing you should consider when you're trying to compare immunotherapy, both SLIT and SKIT, is these are disease-modifying treatments, and their fullest effects may not be appreciated until years after discontinuation. And when I say fullest effect, I'm talking about the cost-effectiveness of the treatment, the potential prophylactic preventive effect of the treatment. And you're not necessarily going to capture that during a one-year clinical trial. Does that make sense to those of you in training, fellows?
Yeah. Yeah.
It's an important difference, because when you start looking at some of the critiques, and there is a recent systematic review I am going to come to, that's kind of saying it's not worth it. It's only this much better than the placebo, and sublingual immunotherapy is not worth it. But some of these analyses don't factor in the disease-modifying potential. And the other important thing, I think, as you are soon to be clinicians out in practice, is I really think patient preference is important. And in discussions with patients about decisions with treatment, some of these factors, such as disease modifying, you may get clinical benefits years after discontinuation, should be included, as well as the risk and benefits during treatment. This is actually a systematic review that compared subcutaneous immunotherapy with pharmacotherapy. So it reviewed 5 different meta-analyses. And you can see these graphs. They're showing you the magnitude of improvement in terms of total symptom scores with subcutaneous immunotherapy and various medications used for allergic rhinitis, desloratadine, montelukast, and mometasone. And as you can see, of the 3 comparator treatments, only mometasone approaches subcutaneous immunotherapy in terms of the magnitude of improvement. And again, this is not direct comparisons, but taking separate studies and analyzing them. And this was another study that looked at different allergy rhinitis medications and the magnitude of improvement over placebo. And you can see the range is 8 to 15%, or 5 to 15%. Now, this particular slide looks at the recent tablet studies, the grass, Timothy grass, the 5-grass, and the ragweed tablet. And to your right, on your screen, are you seeing this little GoToMeeting logo? Maybe I should minimize that. Is that just showing up on my screen?
It just shows on yours.
Oh, okay, but it's not affecting yours. Okay, okay, that's fine. So the far right, you have symptom medication scores, and you can do a quick eyeball, and you're seeing 28%, 31%, 27% over placebo. So you can already see that the magnitude of improvement is at least comparable to inhaled nasal steroids. Does that make sense? At least, and that's pretty consistent with the more recent clinical trials with the tablet products. And these are large clinical trials for an immunotherapy trial. They'll have 600 patients, multi-city, multi-center. And if you look back at the older studies with subcutaneous, you just didn't see large clinical trials like that. They're rather expensive. You have to include patients months before, because you have to start treatment months before, whether it's SLIT or SCIT. And then you have to cross your fingers and hope you have some rain during grass pollen season, so there's actually some grass pollen, because with the natural season exposure, if you have a poor pollen, that has happened to. Okay, now my slide's not advancing. What's going on here? Yeah. Oh, okay. Oh, I'm sorry.
In big pictures and seconds.
Sorry, I thought I went back to the beginning. So I'm going to move on and compare SLIT and SCIT. And this is another thing that I've been hearing for a couple years, and I see it in some of the print kind of news articles comparing SCIT and SLIT. And this is kind of my preview thought. They're different treatment regimens, and they require different commitments of the patient. Subcutaneous immunotherapy requires the patient to come to the office, receive their treatment in the office, and wait. So there's a time component. SLIT requires the patient to be consistent and taking their treatment regularly at home, so there's a convenience component. But it's, for the most part, daily treatment versus SCIT. You get to monthly. So, I mean, these are all things that need to be considered. And some background on subcutaneous immunotherapy, which has been around for over 100 years, we still only have 2 to 6% or less than 10% of patients in Europe and U.S. that receive subcutaneous immunotherapy. So the reality is, there are only a limited amount of patients who might benefit from this disease-modifying treatment that are receiving it, and probably inconvenience is one of the issues that prevents SCIT from being more widely used. So I think when comparing the two, we can do side-by-side efficacy comparisons, but we have to consider when we're sitting down and talking with the patient, some of this discussion will take up. They'll say, well, SCIT, yeah, maybe it's 10% better, but I'll never come into the office and get it regularly. There, however, have been very few direct comparisons looking at SLIT versus SCIT. There have been quite a few meta-analysis, and this is what this slide highlights. Although it's a few years dated, there have been some published since this practical paper. But for the most part, you'll see with SCIT, it seems to have a greater magnitude of improvement. This is standardized mean difference between the treatment and placebo group, and the higher the number, the greater the effect, or the higher the negative number. Whereas with SLIT, you see maybe more moderate effect, although these numbers have a bit of a range. When you look at different systematic reviews that have been recently published, they all seem to have a bit of slant favoring subcutaneous immunotherapy. In some parameter, whether it's immunologic, whether it's asthma symptoms, when they're looking at asthma and rhinitis. But they all seem to have a slant favoring sublingual immunotherapy in terms of greater effectiveness. This is the systematic review that I had referred to in the beginning, and this is in JAMA. It's still in press. It's not in print yet. And the author's conclusion was finding a small benefit of the grass pollen sublingual tablets in reducing symptoms and decreasing the use of symptomatic medications, antihistamine steroids, in patients with seasonal allergic rhinitis. Considering the low magnitude of benefit, the convenience and easy administration do not seem to be sufficient reasons for the choice of SLIT. So this is a rather negative systematic review that felt that the small treatment effect, and there have been a number of letters submitted critiquing this paper that have not yet been accepted for publication, including mine. One of the flaws with this particular systematic review is out of the 13 papers, they included 2 tablet studies that did not follow the protocol that is in the package insert. And they were both with the 5 grass pollen tablet. One used a 3 times a week dosing, and the other used 100 IR, which is a lower dose than the package insert. So that is a recently published. You should be aware of this because it is in a very high-profile journal, JAMA Internal Medicine. And in terms of individual studies to date, this Nietzsche study, 2004, is still the best designed study that compared SLIT with SCIT. It was double-blind, double-dummy, a 2-year study with a baseline year, and they compared change from baseline. The problem is, as with the 2-year study, as expected, there was a fair amount of dropout by year 3. So there was no statistical difference in terms of treatment effect for SLIT versus SCIT, and both showed significant improvement compared to placebo. You can see on the top of the slide is a fairly big difference in terms of the dose used for sublingual immunotherapy, about 200 times the SCIT dose. And that's something to keep in mind in terms of treatment costs in the future. But there was a greater magnitude of improvement in the SCIT versus the SLIT compared to placebo, both in symptom and medication scores, but it did not reach statistical significance. And these other studies in this particular table have also compared SLIT versus SCIT, but not in such a rigorous fashion as the double-blind, double-dummy. And this is just an interesting study that looked at comparing 3 treatment arms, SLIT alone, SCIT alone, and then SCIT buildup followed by SLIT maintenance. And what they found in this particular study is that with the 2 groups that got SCIT, there was a decrease in asthma attacks and inhaled steroids compared with baseline earlier in the treatment 4 months versus the SLIT group, where they only saw it at the 12-month point. They also saw improvement in the rhinitis symptom visual analog score in the SCIT plus SLIT group. And then there were also changes in the regulatory cytokines in both groups, but there were some differences in onset of action. And a greater rise in antigen-specific IgG4 in the SCIT. And that's been seen in other studies as well. And I've just presented that to you. If you're really interested in looking at comparing SCIT and SLIT, it would be worth reviewing Dr. Nelson's paper in Practical last year. And he reviewed the 11 randomized studies that were published, 4 of which were double-blind, double-blind placebo control, and 7 were open but randomized. And in these particular studies, you can see that there is, in both the randomized and double-blind, there were studies where SCIT was the only arm that showed efficacy over placebo. And again, a greater magnitude of effect with subcutaneous immunotherapy versus SLIT. But his conclusion was the relative clinical efficacy remains to be defined of SCIT and SLIT when each is compared with placebo in individual studies and meta-analysis suggests greater efficacy of SCIT. In the limited number of head-to-head studies, SCIT has provided greater clinical and immunologic responses. However, head-to-head studies with well-defined doses by the two routes are urgently needed. And lastly, I'm going to just present this because it's an excellent health technology assessment, a systematic review of SLIT and SCIT, looking at efficacy, safety, but also at economic evaluations. And what their goal was to update the most recent Cochrane reviews on SLIT and SCIT. So those were the studies they included. And then for economic evaluations, they looked at any study that included information about cost. And it's a whole lecture unto itself about how these economic evaluations, what types, how these are analyzed. But I just included this slide if you were interested. If you are interested in this sort of topic area, you might want to read a little further. This document you can actually download. It's a very comprehensive document, but the economic studies can vary. They can have real-life outcomes. Those are generally claims analysis. Then there's different ways of doing cost analysis, and they sometimes assign a cost value to improved quality of life and how much quality of life years, that's QALF, and how much cost there is to provide that extra quality of life year. In thinking about it for allergic rhinitis, it's a little bit more challenging to kind of understand you know, how a treatment improving a quality of life year is calculated, but you would probably understand it better for like a cancer treatment. For instance, a certain treatment gives you 3 more years, but the quality of life of those 3 years is only 25% of what it was. And that's kind of what the concept with the quality of life concept, but it has been determined and calculated, and it's fairly commonly used in economic evaluations of treatment. And it's actually becoming increasingly more important. Payers, government decision makers for healthcare costs are interested in cost effectiveness or comparative effectiveness. The good news is this particular systematic review concluded that both SLIT and SCIT can become cost-effective for quality of life years from about 6 years after treatment initiation. So this is where you're seeing the benefits after treatment discontinuation, because it's assumed that you're looking at about a 3-year treatment period, and then 3 years out, you're starting to see the benefits, and that's probably because the treatment costs, the benefits are catching up with the treatment costs. They do note that these estimates were based on limited data and the use of a number of assumptions, but they also noted that the potential cost savings resulting from future cases of asthma avoided were not included in the analysis, and this would lead to an increased cost effectiveness if these were included. So this systematic review says immunotherapy is cost effective. If you go and read the text itself, it says SCIT might be cost effective at the 5-year point. And if you go look at individual studies, you can actually find individual small studies that have shown cost effectiveness during treatment. One study looked at a 3-year treatment period and then looked at patients 3 years afterwards and found an 80% reduction at the 3-year point that was maintained at the 6-year point. This is another systematic review I participated in with Cheryl Hankin, who's a healthcare outcomes researcher, and she found 24 papers that provided cost information about SLIT or SCIT, and that 23 of these demonstrated compelling evidence for cost savings of both AIT or SLIT. And then in 6 studies that compared cost outcomes of SLIT to SCIT, the preponderance was in favor of SLIT. And those are some of the more recent studies. Again, using some of these assumed outcomes like prevention of asthma. So I'm going to move on from systematic reviews comparing SLIT and SCIT, and again reinforce what I said in the beginning. I think that if one's, say, 50% effective and the other's 40 in terms of side-by-side efficacy, the other factors need to be considered as well. And again, patient preference, convenience to patient, likelihood patient will adhere, and they may be different for the different treatments, and that should not be that should be considered seriously when having a discussion with the patient. And we'll talk a little bit more when we get to safety about the different issues that will require patient safety, and a little bit different regimen that will have to be factored into consideration. But I'm going to move on to a question of, can you glean the effective dose of sublingual immunotherapy from the literature if that allergen has not been studied? In other words, subcutaneous immunotherapy, it appears across various different allergens in studies across the Atlantic and other parts of the world. You come into a dosing range that's pretty tight, 5 to 20 micrograms of the major allergen. Is that the same with SLIT? Can you take some studies of grass tablet and then pick up extract liquid in the U.S. and calculate that dose and administer it? Are you assured of efficacy? And the quick answer to that is no. Each formulation really needs to do its own dosing efficacy regimen. And this is a filler slide, but I had the honor of giving the William Franklin talk in New England Allergy, and he attended, and before I even spoke, he rattled off and got me rattled about, is there a true placebo in these SLIT trials? It's about, the placebo effect, and so the truth is with these studies, there really isn't a true placebo because you can't evoke the symptoms of active treatment underneath the tongue. It's been tried with histamine, so the treatment group probably knows they're getting treatment, although there have been post-hoc analysis that have looked at those groups and active versus the ones who had local reactions versus the ones that did not, and it did not seem to affect outcome. But anyway, this is a slide just showing you the doses that have been shown to be effective with subcutaneous immunotherapy, and again, it's a fairly tight range. This is how effective dose was established, and I think it's important for allergists in training to know the basis of our dogma. And if you look at the dose response studies that have established effective dose for dust mite or grass, there's maybe one for each, and this is the dust mite study, and it compared 3 doses of dust mite. Symptom medication scores are monitored, but they were so optimally controlled, there wasn't a difference per se, and the outcome they looked at was bronchial challenge with dust mite. And based on the bronchial challenge results and the incidence of systemic reactions, they determined 7 micrograms was the optimal dose for dust mite. So that's the dose response for one of our major subcutaneous allergens. In the beginning, when I first got involved with looking at the SLIT literature, and Jay was actually on the work group, as well as Hal Nelson, and we drafted our first draft in Aspen, Colorado, because that's where Hal Nelson wanted us to meet and write our paper. But at the time, there were about 105 articles, and SLIT was about 20 years old. And what we found after looking at all the articles building evidence tables is about 1/3 of the studies did not demonstrate efficacy in the first year, and about 1/3 did demonstrate efficacy in both symptom and medication scores. And the way we built our tables were based on dose and duration and color-coded it to try to step back and look at it. Okay, so where's the dosing range that we're seeing efficacy? And we really could not find that. There was well over a 300-fold difference in the different effective doses that we were seeing in these collective studies. So our conclusion at that time was each formulation needs to do its own safety dosing regimen. And I use this slide as an example of looking at the same allergen and looking at it 2 formulations, tablet versus extract solution. So pretty consistently, grass tablets with 2 different manufacturers have come in a 15 to 25 micrograms given daily, and essentially that's the only regimen that was studied, daily dosing. So we don't know if every other day would have been effective, but that's the dosing regimen demonstrated to be effective. And then in the extract grass solution studies, there's been efficacy shown somewhere in the 10 to 40 and lack of efficacy in that range as well. Dust mites, the tablet studies from Allergens is running about 148 micrograms in the tablet of the mixed mites. And then for solution studies, very limited studies in U.S., and they were not really dose response studies. The 4,200 AU dose was compared against a much lower dose, but it was such a small group at the end of the study, they really couldn't make statistical determinations based on symptom scores. They used 2 objective parameters. And then for ragweed, the 12 microgram tablet was the tablet dose that was approved. They did look at higher doses, but they started seeing systemic reactions. And in their phase 1 study did not go beyond 50 micrograms because of the reactions they were seeing. In the solution study that was phase 3 with Greer, 50 micrograms is essentially the dose that was found to be effective. And it was compared against in an earlier study about 4.8. So if you were like picking up a bottle of ragweed and looking at a tablet saying, okay, I'll calculate what 12 micrograms is, what I'm trying to say is I don't think you've got enough in the collective literature to determine what you would use for a U.S. licensed extract. You have just these very limited small studies which are suggesting about the dose we give. Well, not really, because the dust mite dose is considerably higher at 4,200 units daily than we give in a month. We give about 7 micrograms a month. There is one study just recently that compared the same company's product as a solution and as a tablet. This is ALK's product. It was randomized, and they looked at a number of what we would normally call secondary outcomes, immunologic changes, facilitated antigen presentation, basophil activation, and nasal challenge. And they did see both groups had a significant improvement, favorable improvement compared to control. But again, there were differences in terms of onset of improvement between the 2 groups, with SCIP being earlier onset. So in conclusion, there are no double-blind placebo-controlled studies comparing the same allergen extract administered as a tablet or liquid intra or intra-allergen extract manufacturers. So we don't necessarily know that a 20 microgram AMBA1 extract is going to have a similar performance as a 20 microgram AMBA1 tablet. So until further I notice, I think you still have to have some studies that demonstrate the effectiveness of a particular product that you're going to use. And I think that's the reason I'm kind of hammering this home, is I know there's a fair amount of off-label use within our specialty and outside using, and there's nothing wrong with off-label use, but in terms of trying to determine dosing, it really would require a considerable amount of thought. I know Florida Allergy did this, or tried to do this with oak tree, and they kind of set up a sort of a informal study, and I don't know what the results were, but they decided to go with the equivalent of what we would give in a monthly dose, giving it daily. So any questions? Kind of an important subject area, because I do get asked a lot of questions about this, what to mix with what, and, you know, can you put 3 pollens together, stuff like that. All right, these are the 3 products that were approved by the FDA. This is what their package insert looks like. Each one has a different age range, and that's really based on the data that was presented to the FDA and what they decided they could approve from its safety standpoint. For instance, Orlair did not have enough data in the under 10-year-olds to get down to a 5-year-old. Ragatech wasn't studied in the adolescent population. And you can see the 3 products have different excipients, inactive ingredients. The 2 Merck products have a fish gelatin as their inactive ingredient, and the Allergen product has mannitol and lactose. They also have different schedules in a way. It's pre-co-seasonal for the ragweed and the Orlair product, but the Grassatech product, which is the Timothy grass product, has the option of year-round or pre-co-seasonal. This is just what I said earlier about the, inability to look at the literature and say, I'm going to use a grass pollen extract, and what does the literature tell me in terms of what effective dose to use? And you can see that there's nothing consistent here. There's this one study, and it's a unique product of a 6 grass pollen that showed efficacy, but it was not dose response. A 9 microgram study that only saw efficacy in a subgroup that had the worst symptoms. So this is just, one of the problems with trying to determine a dose of a product that hasn't been studied. There have been 2 studies in the U.S., again, small studies, as I mentioned, that have used a dose of 2,800 BAU for Timothy. The bottle is labeled 100,000 BAU, just to give you some perspective. And one was a study looking at multi-allergen Timothy plus Timothy alone. And the Timothy alone group did demonstrate efficacy in all the secondary parameters that were looked at. And the other one was a study looking at dual allergen immunotherapy, dust mite and grass, and looked at several immunologic parameters and nasal provocation skin testing, and did show efficacy at that dose. I did some number crunching. If you were to try to use that dose daily with now it's 2013 prices, it would cost extract-wise about $7 a month. And these are the ragweed studies, the Schoner study and the Critico studies are the ragweed solution product. The first study compared 2 doses and overall 15% reduction in symptoms, medication scores, which was not considered significant. The benchmark at that time was thought to be 20%. At the time, nobody actually really knew what the FDA was going to require. Now we know it's 15% with the confidence interval between the placebo and the treatment being 10% upper limit. So they went on and studied 50 micrograms with, I presume they thought, a better selected patient population. And in this study, they did show a 43% reduction in combined scores compared with baseline. So that's a little different from what I've been showing you. You're comparing their baseline versus comparing the 2 treatment groups, the difference in the 2 treatment groups scored, placebo versus treatment. And if you looked at that, it's somewhere in the 20-some%. Dust mite all over the map with the solution studies with just a small, less than 1 microgram to over 4,000 micrograms given monthly. And now we have 2 products in development, dust mite tablet products. This particular study at the bottom is the Allergen product, which is labeled in IR units, but the 500 IR has approximately 148 micrograms of the mixed dust mites. And then more recently, Merck has published several papers looking at their dust mite tablet, which is in a unit called sub-Q, and in later studies it becomes DU, but there's no information about the micrograms of dust mites that correlate with these units in the studies. they point out that because we're not consistently using a standardized assay to measure allergen, and there are a lot of pitfalls with the lack of having a standardized assay to measure the allergen content. And one of the arguments for not reporting in major allergen units is that we don't have, other than AMB1 and FeLD1. So this is a study that looked at asthmatics, and the primary outcome was inhaled steroid dose, and they did achieve significance in terms of reduction in inhaled steroid dose, 42% versus 15%, but there were no significant differences in the other observed asthma parameters. Then they went on in that study to look at the group that also had allergic rhinitis and who had some symptoms, a symptom score greater than 1 in the baseline period, and followed them. And they did show that again, a dose response with the 6 sub-2 group having 28% improvement. And then they also studied this, and now the unitage is being used. The language is DU, and this is a challenge chamber study. And when you're looking at a perennial allergen study, it's very challenging for the investigators because they really don't have any control or knowledge of what the real exposure to the patient is. So there's an attraction to a challenge chamber study in that you bring the patient in, you know what their baseline symptoms are, you know that you're challenging them to the allergen. And so there is a growing acceptance that environmental chamber challenges may be acceptable for at least phase 2 of an allergen immunotherapy product development. So this just demonstrates that there's an earlier onset and a greater magnitude of improvement in the higher dose EU product, and these are in nasal and ocular symptoms. So I believe that this is the dose that the Merck is going to move forward with, the 12 DU. And we'll be seeing more studies. I know there are several clinical trials in progress. I heard rumor that they may be shooting for registration in Europe next year, and I'm not sure about FDA approval here. So in summary, I showed you what the tablet products look like. There has been the 2 studies that I've mentioned so far that have looked at a U.S. licensed extract, and one used the dose of 4,200 AUs given daily. This was conducted by Bob Bush and funded by the CAM, Complementary Alternative Medicine arm of NIH. And again, the problem was there was a fair amount of dropout, or not dropout, fair amount, low number of completers. But there were differences in the bronchial challenge and the specific IgG4 group in the higher dose compared with placebo. And then the other study was conducted at Stanford, Carrie Naidoo's group, and they looked at 28 AUs along with the GRASP given together but as separate droppers. And they were able to demonstrate efficacy in the skin test and immunologic parameters. I calculated what this dose would cost if you were to give, it would be 0.28 ml daily, which is about the maximum dose you can give underneath the tongue. And this is, again, is 2013 prices, and it would be about $121 a month. Cat and dog, effective dose of neither have been established. There have been a few cat studies. The dosing range is huge. What was effective and what wasn't effective. Nelson's study at 40 micrograms daily did not show efficacy. These are both done in a cat challenge study. It was not a chamber, it was a room, but that they both used the same tool to assess outcomes. Dog, to my knowledge, has still never been studied. And there was one study in U.S. conducted by a company that I don't think is still in existence, but I don't know who bought it or owns the license. But they looked at a challenge chamber and compared 2 doses, 0.48 and 4.8, and they never released the results. I heard they were not favorable. Cockroach, Bob Wood looked at this as sublingual. He did some biomarker studies. He did different doses and found that there just wasn't a consistent difference in the changes in biomarkers expected with immunotherapy, and they abandoned the sublingual approach for cockroach. I honestly, I say this all the time when I speak, but I just don't think I would have a patient would ever allow a cockroach extract underneath their tongue. So, but you know, this is Florida. And then lastly, one study using a very low dose of Alternaria, a European study, found effectiveness. It was a double-blind, placebo-controlled study using about 6 micrograms a month. And if you're curious about how much all A1 is in our various U.S. licensed extracts, this is an old paper by Martin Chapman's group looking at various companies at 2 time points. But you can see not much. This is 6 micrograms per ml. So to get the dose that you would want for subcutaneous, you'd give 1 ml of undiluted Alternaria. It's just food for thought. And lastly, trees, really we don't have. The tree that's been most studied because it's an important allergen in Europe is birch. But mostly these are being studied in proprietary units. There's a little challenge in converting it over to a language you know from the bottles, you know, 1 to 10 weight per volume or P&Us that we have in the U.S. Oak tree, to my knowledge, has not been studied. And again, I did a cost calculation. If you were to use practice parameter dosing and give it daily for these non-standardized pollens, it'd be about $13, $14 a month. And we are multi-allergen prescribers in the U.S. It's pretty much the rule. This is a very old paper done by Bob Esch where they looked at the average number of allergens in a prescription because they do some mixing for allergists, and we had changed from 10 to 8 in 1999 to 2002. And almost all the studies we have looked at, with the exception of the Nelson Amir study and Carrie Nadeau studies, have looked at single allergens. There have been a few studies that looked at multi-allergen and safety and found there were no safety issues in an adult pediatric population. I showed you the dual study that was done in Stanford, did show that dust mite and grass administered separately did achieve effectiveness. And then Hal Nelson's study, which looked at Timothy versus Timothy and 9 other allergens, suggested that the clinical efficacy may be reduced with the addition of multiple allergens. And I'm going to whiz through these slides. You have them, you can study them. These are the different studies that I just mentioned. Because I want to move over to safety. And this is just a summary of potential costs that I put in a paper for allergy asthma proceedings at the calculations that I have just mentioned if you're interested. But again, the prices have gone up since 2013, so they've probably even gotten higher. In terms of when to start, I mentioned that each of the tablet products has a slightly different recommendation. Studies have looked at different time points, and there have been a few studies that looked at pre versus co versus pre-co versus perennial. And studies suggest, this is a chamber challenge, that you start to see clinical efficacy. Now again, this isn't a chamber challenge, so it's a very controlled setting, at least as early as 30 days. This is a grass pollen chamber challenge. And this is the study I showed you, the Nolte study looking at the dust mite tablet products, and they are starting to see a difference at 2 months, week 8. And this particular study looked at starting the allergen in the beginning of the pollen season. This was a slit solution study in grass pollen, and they started it just at the onset of season, got them up to maintenance on the same day, and they showed a significant improvement in symptoms year 1 that was cumulative and progressive through the 3-year treatment period. And this study looked at 2 months versus 4 months grass pollen tablet, and they found similar efficacy. So this would be 8 weeks versus 16 weeks with the 2 on 2 different schedules. So in terms of when you start, there's package insert recommendations, and then you can look at the literature and sort of decide. I would recommend, if you were to decide to start earlier or later, then it's on the package insert, having a conversation with the patient that you are, and then kind of, if they maybe are a little disappointed in their results first year, it may be because of that. Because there might be some questions, you may want to have that discussion with the patient if you're going to start earlier than the recommended 12 weeks or even 16 weeks. I will say that the dosing regimen really has not been studied, but daily it seems to be where we're going to be, because that's what the studies are being done. So we don't know if every other day would be as effective, but daily is the dosing regimen pretty much of all the recent clinical trials. There's argument that that will improve adherence, but that has not been proven. So I'm just going to move and end with safety. Split application reaction, that's the term we're using, but their local reactions are extremely common. 70-ish% of patients in clinical trials, typically they occur within the first week of treatment and then resolve regardless of what you do, whether you dose reduce, give antihistamines, they seem to resolve after the first week of treatment. But they can be alarming to the patient, and patients need to be appropriately educated about these local reactions. Gastrointestinal symptoms without any associated systemic symptoms like wheezing, cough would be considered a local reaction. There have been a few case reports that have been labeled anaphylaxis. No identified risk factors to date, as we have seen with subcutaneous immunotherapy. A few have been reported in people who had systemic reactions with SCIT, but there hasn't been a pattern that's clearly, or it hasn't been systematically studied to show that that is a specific risk factor. There was one case report last year, or no, it was earlier this year in JACI in practice, where a child who was on dust mite solution, SLID, had a systemic reaction, anaphylaxis, it was labeled, after eating dry foods and then taking his SLID. On exam, he had abrasions in the mouth, and it was thought that that enhanced absorption, and that led or set the stage for the systemic reaction. And there have been 2 case reports, one in a GI journal and one of our journals, of eosinophilic esophagitis with pollen sublingual immunotherapy. And there are case reports in the oral immunotherapy for food that are more so than this. But, you know, there's concerns about that. This is just a timeline of one of the grass tablet studies. And the Ds are days, so you can see by day 8 most of the application/local reactions have just subsided. So in terms of education, it's important to provide education to the patient what these reactions are, that they're fairly common, that they'll disappear, so that, you know, they can expect it and they're not alarmed. The World Allergy Organization developed a grading system for local reactions, and it's in JACI, published a few years ago. They basically are graded in terms of the bothersomeness or troublesomeness to the patient, with a reaction that prompts discontinuation being grade 3 or severe. And again, GI reactions are generally considered local unless there is a systemic symptom, and then the reaction moves over to be graded according to the WOW systemic reaction grading system. And in terms of contraindications to sublingual, it's contraindicated in medical conditions that would reduce the patient's ability to survive a systemic reaction or its resultant treatment. And then there's a long list of other contraindications on the package insert that you should at least be aware of, because your patients get a hold of these and read them and may have questions. One of which is a history of any severe systemic allergic reaction, which would include peanut. Local reaction severe after taking any other sublingual immunotherapy, EOE, and then allergy to any of the inactive ingredients. I got a question at one of the regional state society meetings about this, and I honestly I'd been lecturing on FLIT for a while, and I didn't know what the inactive ingredients were, but they asked me about fish allergic patients in the grass tablets. And so I did my homework and contacted the company, and they sent me this this paper. I think it's the only study, but it looked at fish gelatin in fish allergic patients. And gelatin apparently is only a fraction of the 28 milligram tablet. So one study, they looked at gelatin derived from codfish skin and challenged 30 fish allergic patients, and none of them reacted below a dose of 3.6 grams. So they concluded with a 95% certainty that 90% of allergic consumers will not react to ingestion of 3.61 grams. Again, the tablet's 28 milligrams. So hope that answers that question. A perspective, this is a Canada package insert. I recently gave a conference like this to the Canadian fellows, and I wanted, because our package insert really has a lot of warnings, I wanted to kind of see how similar or not similar theirs was, and there were differences. They do have in their list of warnings beta blockers and ACE inhibitors. Our package insert does mention beta blockers and also about 20 other drugs that you have to be concerned about because of their potential interaction with epinephrine. And this is the package insert. It does basically require that the patient be prescribed an epinephrine auto-injector with proper training. It also, and this is probably not a problem for patients or you, that the first dose is recommended in the office with at least a 30-minute wait. And I think that's completely reasonable, because then you're there to assess if the patient has a funny feeling in the mouth, or you can go and assess them. And I've had, at least one of those reactions where I went to assess the patient and was able to reassure that there were no other signs of systemic reaction. If you look at the package insert and look at about the epinephrine, it also lists a lot of different medications, including some they're likely to be on, like the thyroid medication, certain antihistamines. So be aware of that and think about how you're going to have that conversation. The Canadian package insert does not mention epinephrine auto-injectors, nor is it standard care anywhere, nor is it usual care anywhere else in the world, at least not in Europe. And there have been over 1 billion doses administered in nearly 3 decades, and there's been no reported fatalities, with, again, a standard practice being no routine epinephrine auto-injector prescribing. When the package insert came out, this is myself at a meeting in Genoa where we wrote a paper on SLIT. That was published in JACI. And so these are some of the leading international. This is Walter Canonica, Tom Casale, Hal Nelson, Pasalacqua, and Jean Bousquet. So when the package insert came out, I did email Gianni Pasalacqua, and I said, did you see the recommendation on the epinephrine auto-injectors? And his response is, this is quite crazy. In some point, on which scientific basis was this? And wow, this is risky. And the thought was, do you know the effects or the untoward effects of having epinephrine routinely prescribed? So this is something that we as a entity, the U.S., are going to gather experience on this. So I think it's very important to educate the patient on when to use it, and that part of using the epinephrine auto-injector does require a follow-up. I believe it's either you go to the hospital, dial 911, or contact your healthcare provider. So I think it's an important part of patient education, the epinephrine auto-injector use. And then there is a question of how much will this increase costs of the sublingual. Right now, they're pretty pricey. They come in dual packs, even with the coupons. Sometimes it's been pretty pricey for patients. So that's all food for thought. And lastly, patient instructions. Really is not a lot of guidance, either package insert or even in the literature, what to tell patients when they miss a dose. The package insert for the patient medication guide says if they miss more than 1 day, they're to contact the office. Well, if you're the office, what are you supposed to tell the patients? The studies had different protocols. The Greer studies had them contact the office after 1 day, and I'm not sure if they brought them in. The AOK studies allow for 7 days of mistreatment. And again, my European colleagues, when I contacted them, they said they didn't see problems with these gaps in treatment, and that patients didn't have to updose with the tablets. Therefore, why would there be a concern with the gaps in treatment? And the other warning you should be aware of, if there's a wound and inflammation in the mouth, they're to stop treatment until the wound completely heals. There are forms on the Academy's website that are instruction and consent forms that were developed by Tali Epstein and Chris Calabria and a work group that you can download and use for your practice. And I'm going to end there because I think I'm a minute or two over, and I didn't leave room for questions. This is aproch2. It should be in print. I think it's been accepted, but a lot of the things that I've gone over are covered in this, and this again is a joint document of the Academy and Europe in the IACI, and these are some of the leaders. This is Oliver Farr. He's a big immunotherapy outcome, and this is Moises Calderon, David Bernstein, and this is Nikos, past president of the Academy. So I will hush up.
Wow, great. Thank you so much. I think we have time for maybe one question. Anybody have any questions for Dr. Cox?
Yeah, Jay.
Yeah, Brock, did you want
to ask word? Great hearing you again, all this sort of thing.
I was thinking about you, wondering if you were on the call. I was thinking about you when I was doing my laps the other day.
Yeah. Well, something that always worries me about all these studies is how do group studies apply to individual patients? That's one thing. And the other is how accurate are symptom and medication scores? They're really fairly subjective, and there's so many variables. It always bothers me, either of these studies that only show 20 and 30% efficiency, that when there's that big of a placebo effect too. So it's kind of, I don't know. What's your feeling?
I agree with you, and I think a lot of the immunotherapy research world agrees with you that we really don't have a good outcome assessment tool, that the symptom medication scores, we kind of agreed to use them, but they really do have a lot of limitations. For instance, what if two of the symptoms are not even symptoms that bother the patient? You're tracking them, like they don't sneeze. It's very subjective. You got somebody in a nice controlled study setting, so you kind of brought them to a higher level of control, and then you don't have any control over the variables that are supposed to induce their symptoms, like the season, or if it's a perennial allergen. So I think everybody agrees it's not the best tool. The European Academy actually has a task force looking at immunotherapy outcomes. I think it's one of their high priority task forces. People are liking challenge chambers because at least you've got some, but there are a lot of discussions about that, standardizing them, because I attended one of the conferences on challenge chambers at their meeting. Then nobody's agreeing on what surrogate marker to use, IgG, skin test change.
Those surrogate markers also, you have to track someone and see how they actually change too.
Right.
As a control.
And there's not agreement. There's not really agreement about, I was just reviewing an article by Philippe Monnier that's looking at biomarkers for immunotherapy, and I was surprised how far away we've gotten from, I thought we were getting close, and now we're far away again. So a lot more work needs to be done. But I like challenge chambers. At least they're getting exposure to the allergen, and you have some sense of what they're walking in with symptoms, what their baseline, rather than having them come into season and all these other variables happen.
Right. That's tough.
So, have you ever tasted these? Have you tried them?
Yes, Jay. After you told me that you tasted them all, I did indeed do the same thing.
First thing I did when I got my samples is I put it under my tongue to see what it was like, and The grass tech and the ragweed tech both taste like meringue. They're actually not bad. You put them on your tongue, they kind of dissolve right away, and you're just kind of sitting there for a minute, figuring out what to do. For a minute, it's kind of boring. The oral air is a denser tablet that didn't really have much of a flavor at all, and at the end of the minute, it was still there. It hadn't really dissolved, so I wasn't sure what to do with the residual tablet at the end of the minute, so I'm not really sure about that. But I think if you're allergic to the stuff, you probably shouldn't just pop it under your tongue, but I don't have any allergies, so I thought it would be an interesting experiment. We're still having issues with health plans covering it. It's expensive if you don't have your health plan covering it, and people who don't have prescription coverage, it's a prescription drug. They're having a problem. We have a particular problem with ragweed because we're a pediatric hospital, and anyone under 18 is off-label, so it's never covered. That's become a real issue for us. We also don't have people who are just allergic only to grass or only to ragweed. We always want to use both, and then we can't figure out, do you put them both under your tongue at the same time, or do you put one and then wait a minute and then put the other and wait a minute, which is really boring, 2 minutes. How do you address that?
Well, just to address the last thing you said, because there is a study that it probably will be published soon, but it was presented as an abstract at Eastern Allergy, where Merck looked at the ragweed grass tablets in a sequential protocol where they did one in the morning, one in the evening, then they moved over to both given at the same time in the morning, one after the other, and concluded that at least it was safe. So I guess we'll have in the future at least one study that looks at 2 allergens given sequentially back to back. A study that's double-blind placebo controlled, not the earlier studies, which were small studies that specifically look at safety. Of course, then they have to look at efficacy as well. But you're right. If somebody has multiple allergens, you're only going to cover the one you're putting under the tongue. But keep in mind, you really have a large percentage of patients who they will never get to your office or your clinic. They won't. So you're having a conversation with somebody probably about pharmacotherapy versus SLIT. I mean, the SCIT people, you're probably already going to say, well, I'm not going to be able to cover dust mite or this, which is also an important allergen, and they'll make that decision. Yes, I can commit to SCIT, but it's probably the population that would say, no, there's no way I can do it, but I really have a bad grass season or a ragweed season. You know what I'm saying? It's a population that right now you're probably only treating with medication.
Sure. Okay. Well, it's an exciting new field. Obviously, there's still a lot more to learn, but you certainly illuminated us on many aspects of this. So we want to thank you for spending your time with us, and we'll let you go.
All right.
Thanks.